Right, it's my pleasure to introduce Paul Francois, who is at the University of Montreal, correct? Recently
Yes,
moved. Do I get this
yes.
correctly?
Yeah, it's good. I moved. Yes, yes, I moved.
Perfect. And so Paul uses a widespread set of methods to understand development, genetic networks, and he uses physics, mathematics, machine learning. So he's going to give us update on research, and we're all very much looking forward to the talk. Thank you, Paul.
Thank you. Thank you so much for the introduction. Yes, so I'm from Montreal, and as we were discussing, we know about snowstorms. So it's certainly not the first time. It's not the first time and not the last time that I guess my travel schedule is perturbed by snow. So I'm very happy to be with you today, even though I wish I would be in Boston right now. So I'm going to give you some kind of, so even though that was supposed to be a research talk, I mean, it's still going to be a research talk. I wanted to give you a kind of like also tutorial form of research talk. So you will see, I will tell you about my work, of course, but also about other people's work. And so you see the title, Waddington Landscape in the Age of Machine Learning. I will try to tell you a little bit about things that people are trying to do to look at this problem, which is what I'm going to introduce you to now, which is that we are living a fantastic age where we do have all kind of data coming in, and then we try to, and by data I mean of course biological data like single cell RNA-seq data, or all kind of like today we saw examples. We have those fantastic movies where we can monitor cells, and so then the idea is trying to understand what happened there. And so in an ideal world, well, you would have the data, and so of course we are used to math model, but recently we start having more machine learning techniques helping us analyze this data. And then the idea is that we from that we can start to do stuff with it. So we could do predictions, or if you're a bit more like theory minded or principle minded, you would like to have grandiose theory principles of biology in general. And so the challenge for us as people who are a bit more, I would say mathematically minded, it's what is how to deal with this, how to get from data to really mathematical models. And as you're probably aware of, it's quite challenging. And I'm going to describe a few changes. By the way, feel free to interrupt. I'm supposed to speak for 45 minutes. I'm very happy to be interrupted by any question. My talk should not go further than that. Anyway, so what are the challenges? And so I think one of the very first challenge we have is that we are human being, and so most people try to build representation of their data, and why we want to build representation data is because we want to get some kind of intuitive sense of what is happening and to do some theory. And so the question, the first question we can ask is that can we build a good geometry or good representation with the goal of building a good theory of what is happening in this complex biological system. And so if you're a bit familiar with this field, I don't know, I don't know, I guess it's a rather mixed audience, but like there are standard techniques like UMAP that people are using to essentially plot this high dimensional data such as single cell RNA-seq. But then UMAP is a kind of like it's very mathematically complex and interesting, but it creates some issues. So for instance, you have hyperparameters in UMAP, and then if you change a bit the hyperparameters, you basically change the structure of the data. So this is the same data set. If you change a bit the parameters, the topology of the representation is changing. So for instance, here you have like kind of like point at the intersection between these three domains, but here it's completely disappears. Of course, you have this very famous curse of dimensionality. You go from say 3D to 2D. UMAP is essentially destroying the structure, and then there are like a lot of debate. If you're interested, if you want to have a bit of a controversial a sense of the controversies, there was this paper published a couple of years ago explaining that essentially why you should not use UMAP because there is a all kind of problems, statistical problems to represent data. So that the first question is really this, how to represent data. And it's very complicated. But then even if you have a good representation of the data and a complete representation of the data, there are other issues, and I'm going to illustrate the issue, I'm going to use a physics example that I'm going to call the virtual solar system for reasons, I don't know, like those days we talk a lot about virtual cells. And so what I'm going to show you, the exact same kind of data in two ways. The way we plotted them in before 1543 and after 1543. And so this is the planetary motion. And so before 1543, we had these very nice epicycles to describe the planetary motion, and we even had very nice models to describe them. So there are these machines that were designed to, predict eclipses and everything, and they were very predictive. They were very useful. So that was working. But of course, now we have a better way to understand the data. Again, this is the exact same data, is just plotted in a different way. And from that we could get Newton's law, and so we really want if we want to understand biological data, of course we want to be more on the left side. We really would like to have some kind of from the data we also want to extract some concepts with the idea of building theory. And if you have a good theory, you can even go further. Like for instance, imagine you have those data and then you see, oh, there's an issue with Mercury's perihelion precession. And then, on the right, you just add another epicycle to explain the data. And on the left, you just say, well, actually, maybe there's something wrong in the theory. And then you discover general relativity. So that's also another reason why you want to get a good theory, because, you might really build up and get more and more precise mathematical representation of what is happening. Okay, so this is very broad so far, and then I'm going to focus now a little bit on biology, and in particular, how we try to understand development. And so this morning we had a very nice talk by Mattia, telling you about mostly about mechanics and flows and morphogenes and positional information. Now I'm going to not to tell you a lot about that. I'm going to tell you about how essentially gene networks inside the cell. So this would be related to development, but the goal here is to try to understand how the gene dynamics within the cell, somehow combine to give you interesting phenomena. And so the general hypothesis, during development, say, you have all kind of gene networks that are acting, and then you have all kind of dynamics occurring. And then the general idea to describe this is that somewhere, in this very complicated pattern, in this very complicated space of gene expression, you can extract there is some kind of low dimensional space where you can describe the data. And in this low dimensional space, you can describe things like flow. And then, you might be able to do a mathematical theory of the flows in this space. And so that's kind of what we call the manifold hypothesis. Is a bit a kind of jargonic term that people use also in machine learning to say, even though the data are very complicated, you can somehow reduce the dimensionality and get a sense of what is happening here. And so that's really what we want to do. And then the question is not only to build, a representation of the data, but we also would like to have, some kind of vector field to represent how the cells are moving in this genetic space. So, for instance, this is the kind of thing that people are trying to do. You have this representation of, single cell RNA-seq data. So each point here is going to be one cell, and then you try to connect the different tissue, the different cell types as you differentiate. And you like to construct such a map, and then clearly when you look at a figure like this, it's really challenging because you have all these phases. It's not clear how to put them on a 2D representation. Maybe it's not even possible to put them on a 2D representation. So this is kind of really the challenge I'm going to try to discuss and address with you today. Okay, so that was pretty, general introduction, and the guide. One of the guide of this is a relatively old theory now that is kind of coming back those days to somehow describe those dynamics. So this is a famous Waddington epigenetic landscape. So I don't know if I am the first to show Waddington landscape in this conference. I guess it's only the second day. I don't know if someone showed it yesterday. It will come at some point. So am I the first? Do I get? Okay. So, Waddington epigenetic landscape. So this is the idea that to describe development, you have a so-called landscape. For those of you who don't know about it, and then essentially you could represent, say, the state of a cell by a bead or whatever, a ball, kind of rolling down this landscape. And then these different attractors that you see, the different values will correspond to different cellular states. And then, based on signal, you will kind of bifurcate and go one way or another. So that's basically a very coarse-grained caricature dynamic model of the dynamics that cell can follow. But it's really it's coming back for all kind of reasons those days. And one of the reasons is because I think people have really tried to understand what is happening in gene networks for a few years now. And it's not that it's impossible, but we need somehow some kind of higher level representation of what is happening. And I just pulled out this review to illustrate that essentially at the systems level, thinking in terms of landscape might be the right way to think about all kind of things, all kind of developmental transition, including maybe possibilities. Okay, so this theory is coming back now, and one of the reasons why it's coming back is because there were also like kind of like new theoretical results, or maybe it's not so new, but more like refurbished, or I don't know exactly how to describe it, but essentially a renewal in interest coming from essentially the data on the one hand, and then how the data could be explained mathematically. And so this is two papers that were published those last years by people like David Rand, Eric Sigia, also James Briscoe, where people really try to explain data with landscape, with this Waddington landscape. They really show that you could very nicely describe and predict the dynamics of cells with a landscape. And what I mean by landscape, it's really a landscape. It's really like an energy function like this. And then, you kind of start to somehow modulate this landscape, and you can really describe what happens quite accurately in terms of differentiation and cellular fates, and predict what is happening. Okay, so I'm going to tell you about that today. One thing that is interesting that those people somehow suggested, demonstrated that even if you're very high dimension, as soon as you assume there is a low dimensional hypothesis somewhere, there are some kind of generic things that you expect. And so if biology follows math, you should be able to see those generic things and understand how things like differentiation is working. So the question I'm going to ask today more specifically is there a way to systematically find such landscape? And I'm going to ask two questions, because it's very vague questions. So the first question is that imagine you don't have that much data, or imagine you really like consider some kind of abstract model of development, and you will see what I mean by this. What are the typical landscape you might expect? And then another question, of course, is like imagine you have data. How can you build a landscape from data? And so those are two questions I'm going to address. Essentially, I'm going to kill two birds with one stone. You will see what I mean by that. But before I move on, I should say this is really like a very active field right now. There are many papers that were published in recent years. I think some of it started with actually Pankaj's meta paper that was some time ago. It's an older paper, very nice paper describing upfield models for differentiation, but now there was a renewal of interest in recent years with all the data coming out where people really try to infer the landscape from data. Okay, so now I'm going to tell you a bit what we've been doing, what we've been trying to do, and to do that, I'm going to start with a kind of a metaphor, and it's also related to some discussion in the field and some standard approach to the field. So I'm going to, I'm from Montreal, Canada, and then I'm going to start with a kind of, oh, Mattia, you have a question?
Yeah, so on this reduction, so if I think about two cells in the gene space, which is in principle very high dimensional, we can think of it maybe in 3D. I can go from one point to another, either on a line or on a surface. So on a surface, I can go from one point to the other in many different ways.
Yeah.
Somehow this framework limits this transition possibility. It's always one dimensional. Is it what is the biological implication of that?
No, it's a good question. Well, I guess you always follow the unstable manifold. That's a bit the idea. But then you're going to try to start adding, So first, it's not entirely true. You would only follow a line because you could have some noise, and you see noise will play a role in some of the things I will show you later. But I agree that kind of in principle, if you're fully deterministic, you will follow lines most of the time. You don't have like some kind of flat things or where you have several trajectories.
Yeah.
So I think the
So yeah,
first
it's I mean it's really the unstable manifold could be very
yeah,
high dimensional, not really one dimensional. But in
so,
this simplification, which I really like, it's always 1D because you really go
yeah.
and I was wondering if this has some biological consequences. I never really understood what is the biological consequences of reducing this to 1D. But maybe we can discuss
Yeah,
offline.
we can discuss that later. But the fact that it's essentially locally 1D and then decision, of course, are also very low dimension. This is what allows you to classify a bit the decision, for instance. So that's a bit the argument that you have very low dimension, and so you can predict how you're going to move in this space. But you're right. I think some of this is coming from just looking at the data, and then people seem to see that those things are mostly one-dimensional, but
Ah,
modulo noise, of course. But I think it's a completely fair question. You could totally imagine that you would get some more complicated things, and sometimes you have several pathways to the same state. This is what is happening sometimes. So that's a bit of a complication, but still locally it would be relatively 1D. But yeah, I mean, we can talk about that later. I will add another kind of decoration later on this story that you might find amusing too later on. Okay, good. Topography versus topology. Why am I mentioning it? Because actually it relates to your question. Many people tend to describe this as essentially trajectories of cell as connection in a landscape. So you're basically going to have saddles, and then you have saddles, and you have trajectories between attractors. So I'm really using this kind of subway map for Montreal, where if you want to go from one point, so let's say as mentioned earlier, I was at McGill, and then I moved to Université de Montréal, and so yeah, there are several pathways, but essentially they are 1D, so I can follow a line, and so this is the way people think about those problems precisely. And then mathematically speaking, you're going to have things like normal forms, and unstable manifold to really describe what is happening and how you're moving in this space. Okay, which is nice. But for all kind of reasons, you might want to do something slightly different. And one of the reasons why you might want to do something different is that if you have data, it might be difficult to actually find those manifolds, the unstable manifolds. It might be difficult to really identify the saddles, the normal, the control parameter and stuff like this. And so, to some extent, what is more intuitive, and also coming back to this landscape idea, is to really think more in terms of actual landscape. And so now, if you have a landscape, of course, let's say here, this is a topographic map of Montreal, where the repeller are, you're going to have a saddle point, literal saddle. And so once you have the landscape, essentially everything is defined. So let's see how it works, and let's assume we have some landscape. And so what we try to do is to actually model explicitly the landscape. And so what is a landscape? A landscape is going to be essentially, a collection of hills and valleys, and then once you have a collection of hills and valleys like this, you can really take your ball rolling and then predict where the ball is going to lead, and then you can really define things like canalized trajectories. You can also say that cell states are actual attractors of dynamics that will correspond to bottom of the valleys here, and then you can have like decision that will be modeled as bifurcation. And one thing I like with landscape is that they're very interpretable, and so you can really immediately know what is happening and go back to experiment potentially, and then validate what you see in terms of trajectory. So
Paul,
mathematically,
can I ask a
yes,
question here?
of course, of
Um,
course.
so this landscape paradigm, is this metaphor maybe too worn out? And the reason I'm asking is increasingly in ecology, people think that it's not a landscape, it's not frozen, it's actually continuously changing.
Oh, I
And and
totally
and the
okay.
question is, Okay, so the reason I asked that is because that means then you would have multiple scales, a scale
Yeah.
over which the landscape is varying and another scale over which fate is changing or whatever you want. Is that
Okay,
something? Yeah.
if I get
Is
there,
that something you're going to address?
if I have time to get there, I will show you stuff about
Perfect.
that at the very end of
Okay,
my
thank
talk.
you.
But I'm not sure I will have time to get there. But definitely we will change the landscape. If this is one of the questions you're asking, we're not going. But then the question of how you change the landscape is a very interesting question too.
Yeah.
So I will show you. Hopefully I will have time to get there. But I start with, let's say for now, not quite frozen, but let's say the landscape is changing very slowly for now. Okay.
Fine.
So
Thank
now,
you.
how am I modeling a landscape? Okay. Well, landscape, I'm going to say again, those are hills and valleys, and then, and so the way we're going to model it, and you see why we do that later, we're just going to assume that we have kind of like a 2D play, a 2D space, and then in 2D space, we're going to define, an energy essentially, and then the energy is going to be to essentially drive the dynamics. Okay. So now, and then we're going to assume that there are some modules, and what I mean by module is really like locations where you're going to have hills and valleys driving the dynamics. So as an illustration, and so to go back to things that have been proposed a few years ago, you can make very simple landscape model of what we now call, I don't know if it's kind of if it's accepted, the canonical binary decision. So Rand and co-worker a few years ago essentially listed what can happen in terms of landscape to get a binary decision in a very specific context, where imagine you have progenitor cells on the top of a tree, and the progenitor cells can give you two different downstream fates. Okay, and they show mathematically that basically you have two broad ways to do that, which they call the double cast landscape and the heteroclinic flip landscape. And so the difference between this, so I'm going to use illustration by my PhD students in my group, Victoria Molczulska, in terms of landscape, is that the differentiation, so double cast landscape, so you basically have something like 3 attractors. And then you're going to have 2 saddles, and essentially 1 saddle between every between pairs of attractors like this. And then the way you do it is that the way you're going to have a differentiation is that essentially imagine you are here. Let's say this saddle is kind of cancelling out with this one, so that you are going to this attractor. Then what happens is that you have a progenitor, you differentiate, and at the same time you differentiate, you actually choose the fate you visit. So it's kind of differentiation and specification at the same time. So that's the double cast landscape. The other one that was proposed is what is called the heteroclinic flip landscape, which is slightly different. And the idea is essentially you separate a little bit differentiation and specification. So this is the case, like you have your progenitor state, and then you have a saddle here, and then you're going to have a cancel. Let's say you cancel out those guys. And now you are committed to go somewhere, but you don't know if you go on the left and the right. It's only a later decision that will bring you to the left and the right. And that's why the heteroclinic flip landscape, you can think about it as differentiation, then specification. And if those things are not clear, I will come back to that later, and I can show you. I have actually some interesting, maybe more intuitive ways to show that later. But by and large, what you can do is that you can, with this process of building little hills and valleys, you can easily reconstruct this canonical decision. It's just a proof of principle that the formalism I'm describing is working. This was proposed in the context of normal forms for using actual energy function, catastrophic theory. But you can really describe those decisions with our ideas of having little hills and valleys, and then just changing a bit the size of the hills and the valleys will give you this differentiation pattern. Okay, so it's just to show you that this works. The thing we're going to add, for a reason that is going to become obvious in 5 minutes, is that we're also going to add rotational elements. So usually landscape are pure energy, of course, but then you cannot have, say, an oscillator if you have a pure landscape. And so what we can do is that we can assume also in this landscape that we're going to have rotational element, and so that will allow us to build rotations, build oscillators, and in biology there are oscillators. That's why you want to do that. And all in all, what we're going to to do is that we're going to assume that we're going to describe the dynamics in this epigenetic landscape by a combination of an energy function, a gradient potential, and a kind of rotational potential that is going to give you local twist. And it's very similar to the Helmholtz decomposition in electromagnetism for the physicists in the audience. And so with that, we can basically describe, we're going to try to describe biological dynamics with this framework and then see what we can get. And so, I mean, there are things we can do. For instance, you can get, of course, classical bifurcations for oscillators, things like Hopf bifurcation or Sneak bifurcation. I'm going to skip that, not spend too much time on that to go directly to the result. And then what we're going to do is that we want to actually build a landscape. And so what we mean by building a landscape, that we're going to combine those elements to essentially try to do things like fitting data or try to explore interesting dynamics. And in the next couple of minutes, I'm going to show you what that means and what kind of landscape you can get for very specific problems. You just need to find a kind of a loss function in machine learning term to optimize the landscape, and then optimize it, and then that will tell you what kind of landscape can describe what kind of data. Okay, so now I'm moving to examples. And the first example I'm going to present is actually, I guess I'm spoiling a bit what Olivier is going to tell tomorrow. The segment, like vertebrate formation, in particular the segmentation clock. So what do I mean by this? So okay, I lost my cursor. Okay, so this is a snake. And this is a very nice paper from Olivier from a bit less than 20 years ago now. Where the vertebrae, a snake of course has many vertebrae, and then the question is how do you form this vertebrae? And what is really striking is that this vertebrae form as the embryo elongates, so you have this very nice pattern of elongation and formation of the vertebrae. And so it was shown by discovered by Olivier and then studied by many people now that the reason, the way you form this vertebrae is that there is as the embryo elongates, so this is a mouse embryo on both sides. As the embryo elongates, you have what is called a segmentation clock. So you have oscillations and waves, and then the waves travel in the anterior, and kind of when they stop, they form essentially boundaries of so-called somites. Somites are the precursors. They're not quite the vertebrae, but they're the precursor of the vertebrae. Okay, so I got interested into that for some time. And so the question now is that can you make a landscape description of a process like this? Okay, and so one way to think about this is that, so let's say you have the embryo that is elongating as a function of time. So this is space, this is the anterior part of the embryo and the posterior part of the embryo. So we try to look at a fictitious gene that would oscillate, and then what is happening in the future somites that there is usually an anterior to posterior pattern that is appearing. So you have a kind of a process transforming a clock into a pattern. And so we actually wrote a review with my student Victoria 2 years ago. If you're interested in models to describe this, so there are many models that people think about. And now this is what we're going to see is that we're going to try to figure out how we can model this with landscape. And so that's a changing landscape to answer one of the questions. So we're going to assume that in the posterior, say, there is one specific landscape, and then in the anterior, there is another landscape. And then the landscape are going to describe the dynamics of your cells. And then the question is what kind of landscape can you get that moving you from posterior to anterior so that you have all this kind of process of patterning that is happening. To do that practically, so I'm going to assume that, so the landscape, we have, again, a posterior landscape and an anterior landscape. And then I'm going to assume that the landscape can change from posterior to anterior, and I'm going to assume that there is some kind of control parameter or a moving sliding morphogene, that could represent all kinds of things like FGF gradients, something like this. And then as the morphogene is passing around the cell, say, or more specifically, as cells are moving, are changing the morphogene concentration, they're going to change the landscape. And then what we're going to do, we're going to assume that as the cells are essentially, feeling this morphogene, this moving morphogene, the dynamics of the landscape is going to change. The landscape is changing, but also the dynamics of the cell are changing. And what we want to do, we want to have something that gives you a pattern in the very end. I'm not telling we get an oscillator or anything. I just want that at the very end of this process, I form a pattern that would be corresponding to the anterior to posterior patterning in somates. Okay, so I'm telling you this just to show you. So when you do that and you use our optimization procedure to actually build landscape, and we get all kind of solutions. And I'm going to show you on the next slide 2 solutions that were found in terms of landscape. And so the first result we have is that it's very, very strong. You know, optimizing the landscape, we get essentially always oscillators, which is really nice. So because we did not explicitly want it to select for oscillators, but you know, it just comes out of our evolutionary procedure, our optimization procedure, and you get oscillators. And so now what you see here, what is happening here is that actually the landscape is changing in time. The change in time is essentially mimicking, you know, the moving of the cell from posterior to anterior. And so initially you have an oscillation, so the cells are oscillating in this gene landscape, and then at some point you have bifurcation happening, and because of this bifurcation, the cells are going to localize in either of these rostral and caudal fates. And then you can do all kind of things. So I'm showing you those 2 examples because they are quite interesting. They give you different bifurcations in terms of the way you move from an oscillator to this binary fate. I don't have time to go too much into the detail of what is happening here, but what is really interesting is that essentially this one on the left is very similar to the dual cusp switch that I was telling you about earlier for binary decision, except that it's an oscillator, and so it's done through what is called a sneak bifurcation. While the one on the right is more similar to the heteroclinic flip with oscillator. So what will happen here is that you are going to have a cycle that is going to disappear through a homoclinic bifurcation. And you can notice here that, for instance, these guys are the future fate of the cells, and then you have a homoclinic bifurcation here, and then the cells are differentiated, and then they decide to go to either of this fate. Oh, sorry, I don't have the right my cursor on the other screen. I'm sorry. Okay, fine. Sorry about that. Anyway, so it's just to show you that, you know, you can optimize this landscape. They can change in time, and then it can give you a nice pattern. And so you can do all kinds of things with this to study what kind of landscape you can expect to explain this kind of wave from posterior to anterior. And there are really interesting results that we get for free. So for instance, if we want to have more segments, you naturally get faster clocks, but you also get bias in terms of bifurcation you get. So we get all kind of interesting results like this. I'm happy to tell you more about that offline, or it's actually published. So you can just check the paper. But you know, you can start running experiments like this to really generate all kind of landscape corresponding to an abstract situation like this, where you want to make a pattern. So that's what I would call a problem where we don't have much data, and you want to explore, you know, what kind of solution, what kind of landscape will give you this nice dynamics. The second example is an example where we have a bit more of data. And so those are data from the Briscoe lab, and we essentially revisited this first paper where the landscape formalism was used by Saez et al. In 2021. And then what they do is they're actually studying neuromesodermal differentiation. And what they do is that they do all kinds of experiments with different morphogenes at different days. And then they're trying to look at, you know, what kind of different fates you have as a function of type, as a function of time for those different experiments. So for instance, in the first protocol where you only have FGF, basically, you know, you have for 3 days you have this fate, and then this pink fate, and then for 4 days you have this purple fate. So this is epiblast and anterior neuron, I think. And then you change the conditions. So say you only put, you leave endogenous FGF, and then as development proceeds, you have this proportion of different phase that is changing as a function of time. And so they have all these kind of experiments with different morphogenes. And then the idea, what we're trying to do is very similar to what they did. It's to see what kind of landscape can give you those data. So in the initial model, they really had this purely topological view, where they were only trying to see, you know, what kind of bifurcation it is and how we can change the control parameter to explain this change of phase. But what we want to do ourselves is really optimizing directly landscape that will explain this. And of course, this landscape will depend on time and by the morphogene that you put. So we did that. And so just to give you a sense, I'm going to show you exactly some specific example, but those are the examples of landscape we can get to essentially explain the data. And we get many landscapes, so that's one of the landscape. And so the idea here is that you have different landscape corresponding to different experimental conditions, and then when you change the morphogene of interest, say Karen of FGF, you're going to change the landscape immediately, and then the cells are going to move differently because of the landscape. And so in the next slide, these are two solutions that were obtained by our optimization process. And so you look from the top, and then the big circle are essentially the attractors. And then you see the trajectories in these landscapes. And so you see, as you change condition, you're essentially changing the landscape, and that influences the flow of the cells. And so if you look carefully, you know, at discrete times, the proportion of cells in every single phase, say yellow, green, or blue, is the same as the experiment, is the same on the left and the right. But clearly, as you see from these movies, you know, you get different results. You know, you can get different qualitatively result flows, qualitatively different landscape. But, you know, eventually, you know, again, we optimize the experiment, so we get the right proportion similar to the experiments at every single time point. Okay, so one of the advantages of our approach is that this is quite easy to run, so we can actually generate all kinds of landscape consistent with data, and then we can start to look at things like, you know, what is typical of this landscape? What is happening on average? And so we did that. So we did this for those experiments, you know, where again you have this multiple conditions, and then we look at the proportion of fates as a function of time. And then what thing that was quite interesting is that, for instance, if you look at
Paul, can I ask a question real quick?
Of course. Please.
Do you optimize over the level of noise too when you do these things, or how do you
Oh,
deal
that's a
with...
great question, Pankaj. Okay, so we could, but we did not optimize the noise. We kept it fixed for these things. But you're right that we could also. I mean, like we just kept it simple. Those data are simple enough that you don't need to go into this direction. But I agree
it's just
that
white. And it's
yeah.
just white noise at some
Just
temperature.
white noise, but it's okay. It's a huge problem. I mean, I'm deep into that right now. Like
Okay.
The noise
Okay.
level is a huge problem. Like, I mean, why are you taking white noise? You know, you could put color noise. You could put also like, anyway, whatever. We can talk
Okay.
about that. Yeah.
Sounds good.
Can I ask a question here, Paul? Sorry, continuing. What is the metric? In addition to optimizing the end state, are you optimizing the cost associated with moving through that? I missed that. Maybe you'd said that. I apologize.
Yeah, I did not tell. We have no cost here. We're just optimizing essentially the Kublai-Leibler divergence between the observed observed.
Okay. So,
yeah.
So is it useful? Is it possible to actually think about this using ideas from optimal control theory, stochastic optimal control theory, where you say that, and you know, we've been playing with this a fair bit recently, and to say that what you're doing is steering through this landscape, but there is a cost associated with steering, and then there is a cost associated with accuracy corresponding to where your terminal fate is.
Yeah. So I mean, one of the referee asked us a question along those lines, which would be
not me.
instead of having a landscape describing, say, the flow, you could say it's actually the probability distribution, and then you could just do, you know, Wasserstein distance between the distribution and optimize that. So definitely you could. That's not what we did, and I think that's an interesting avenue. And that's a question I had for a long time because, for instance, I mean, I basically I agree with you. We should look carefully at this. One thing I find interesting is to think in terms of what kind of bifurcation would be induced by, you know, some kind of optimal transport of, you know, distribution and flows and stuff like that. And I have some ideas, and I've looked a bit on my side on this, at this, but I haven't published anything. But that's definitely a question I'm interested in. Yeah. So I
Sorry
agree.
that you're not here. So, but you're not going to be here next week either? No way?
Yeah, no, I'm sorry. I'm traveling, as I was telling. I would love to talk more, but, you know, I'm happy to talk more about that. And I have thoughts. So okay. Okay, so coming back. Yeah, so the interesting thing we get is if you compare all the landscape solution we got, there is a kind of, of course, you need to adjust for, symmetries and stuff like that, but essentially you can align all the landscape with respect to one another. What's really striking is that the typical topography of landscape we get is rather constrained. So on the left, you basically have a representation of the probability distribution of observing one attractor at a given position. We centered everything on this caudal epiblast attractor. And then what you see is that you have very clear hierarchy of attractors that essentially fits the idea that people have in terms of how the different fates and attractors arrange in this low dimensional representation of the data. So, okay, that might not be surprising, but I think to some extent it's a bit surprising because this is really coming from fitting the proportion of the cell at every single time point. And so, of course, then you could say, oh, there's a different direction in development. And so that makes sense that, epiblast is coming first because this is kind of the origin of development. But still, I find it remarkable that just based on the proportion of fate as a function of time, you get essentially information about the location of every single attractor. I guess, the argument is, if you're moving, if two attractors are kind of expressed at the same time, they're close to one another. So you have implicitly a temporal to spatial map there, but still I find it remarkable that just based on this temporal data, you get something that makes sense. And here, of course, there is no, we identify the phase, but there is no gene or anything. It's just based on, temporal data. Okay, so the second result we got, and of course, now I'm seeing I'm running out of time, so maybe I show very quickly a moving landscape at the very end. But what is really interesting that for the final decision, we actually get two solutions consistent with data, which actually correspond to the two solutions that I showed you earlier, which is a dual cusp and the binary flip. And so this is a case where, I wish I were here, because I actually have a 3D printing models of my little landscape that I can see here. So maybe I'm going to stop my sharing for 1 second. Okay. So you can actually look at the solutions and then 3D print them. And so this is a dual cusp landscape. And I like that because, you can play with it, as Vladimir Arnold was telling, it's kind of like a experimental mathematics, you can have the landscape and then you can imagine the bead rolling here. And what's interesting for the dual cusp landscape that I'm showing you, so imagine you start here, and then what is happening is that imagine that the noise is moving the bead around. Very clearly, you're going to move on either side very quickly and then roll essentially straight to the attractor. So I guess, again, if you have it in hand, you understand it better. But that's the dual cusp landscape, and that's our solution, one of the solutions found for the final decision. The other solution found for the other decision is, essentially the kind of more like standard Waddington landscape, which is the binary flip landscape that you see here. So the bead here is going to roll down, and what is interesting is that as it rolls down, the noise could move you around. And so then you could choose either of the attractor, essentially depending on the noise or like a bias initial condition. And so it's just to show you that, you can get, okay, I'm going to share again my screen. Well, I'm trying to do too complicated things. Okay. Okay, and anyway. Okay, I lost my presentation, interestingly. Okay, good. Good. Okay, so I guess I'm running out of time, so I'm going to wrap up. I guess the last thing I want to say is that, especially for the one thing you see in those two decisions, is that for the dual cusp, you're really going straight, while for the binary flip, you can really kind of move around and have more like a triangular shape. So this is something you would see in the data. Okay, so I guess I'm running out of time. So I hope to convince you that those landscapes can actually be derived even for a very small amount of data, and that gives you some kind of like minimal model of what can happen in differentiation, which I find useful. I was a bit surprised to see that the topography of landscape was much more constrained than what I would have expected, and so again that should be true, with whatever dimension you consider, whatever genes you consider. And then, those things are not ours. It's kind of confirming that things that people have already described, that you can describe the landscape based on the way they change. And I think many aspects can be understood with simpler, smaller system. And what is missing here, the thing that is missing here, is that how you go from the landscape to the actual gene expression data, which is something that we're working on right now. Like we are working on the next generation to really, once you have this kind of latent description of the dynamics, it should be possible to rederive the entire gene patterns and train a decoder of some sort to do that. I mean, otherwise there's something wrong, but if there's something wrong, we're going to learn something. Okay. And just to answer the question I had earlier, we're also working on a much more dynamical ways to describe landscapes. This is a toy model. I was intending to get there, but essentially this is a landscape that is changing in time, and I don't have time to describe what is happening here. But you're going to have cells here, and you're going to see how they split. So we're very much working on this idea that the landscape itself can evolve in time based on all kind of things and signal that you can have between the cells, and then you can see you will see that there is all kind of binary flip and stuff like this. So each of the square here is a cell essentially, and we're working on formalism to explicitly describe changing landscape based on also data. So this is a synthetic model, but we also have data. And I'm going to wrap up now. I would like to thank, so this work, most of the work I've shown you has been done by Wiktoria Moczulska with a grad student in my group, and discussion with many people, including Madhav Mani, Eric Sigia, Alexander Olela. Okay, I guess I'm running out of time, so thank you so much for your attention, and I'm happy to take any questions. I already had questions, so it's okay.